Hello, everybody. Welcome to Virology Live. I'm Vincent Racaniello, and I'll be your professor for this live streamed virology course. I'm doing an experiment, and you're joining me. And the first thing that you can help me with is let me know if my audio is good. This is a brand new setup here at the incubator, made possible, of course, by your donations. All of this new tech you bought. So let me know. This is the incubator, by the way. It is bare bones because you see there's a white wall behind me, but I do have a light, I have a camera, and I have action. Audio is great. Good to hear it. We are going to do an experiment. We're going to see if you can teach a virology course online. I think it will work, but like any other experiments, you have to try it to find out. So we're going to do that over the course of the last lecture of this course will be something like January 3rd. I'll get into some of the nuts and bolts in a moment. But I thought this would be interesting to do. We have a dedicated studio space here in New York City, the incubator on 7th Avenue, and I can come here and do all kinds of science communication. So join me in this experiment. Those of you who have taken my Columbia University virology course will find this familiar, but of course it'll be updated as we go along. And the cool part is that you're here. You're here live. Let's see, we have now 158 people, which almost tops the biggest class I ever had at Columbia University. I once had 170 students. I think we're about to top that. We just did. 175 people are here. So this is cool. I can teach people, and they're all over the world, right? People from all over. You can see in the chat. Holy cow, folks. I am so impressed. I'm honored that you came to listen virology. I think this is a new beginning for teaching online. Very cool. So we have a bunch of moderators here today, of course. We have Steph SF. We have Vanity Nutrition. We have Mr. Ozzycam. We have Les Fabi. And one more who I'm missing here. From Canada. Let's see if I can scroll up. Firuza Lalani. And these are the people who have been here on this live stream that we do Wednesday nights, which is tonight, by the way. And they're helping out. All right. So let us begin here. I have some interesting ways to present material. Now, I'm going to do slides, and I'll take breaks for quizzes and for your questions. And I ask you, of course, to please keep your questions related to the lecture. So let us bring in our slides. I believe I have to switch to my keynote thingy here. Here we go. All right. So this is Virology Live. And for the first session, I don't call them lectures. I purposely didn't. I don't call them lectures because they're sessions. It's different from a lecture. I'm not just standing in front of you. You have the ability to ask a lot of questions in the chat, which is, you can ask questions in a class, but you'd have to interrupt and so forth. So anyway, if you notice the colors of Virology Live, those of you who have seen photographs of the incubator know where they're coming from. So today we're going to talk about what is a virus. And on every session, I have a quotation on every lecture. And this one is particularly important here by Salvador Luria, who wrote the first virology textbook that is based on principles, not viruses. So many textbooks, every chapter is a different virus. You don't learn virology that way, at least not beginning virology, in my opinion. I think the best way is to do it by process, and that's what we're going to do in this course. And Luria wrote there's an intrinsic simplicity of nature, and the ultimate contribution of science resides in the discovery of unifying and simplifying generalizations rather than in the description of isolated situations, in the visualization of simple overall patterns rather than in the analysis of patchworks. So that's what this course is all about. We're going to look at unifying generalizations, and we're going to use a set of viruses to do that. Now, before we move into the material, I want to give you some contact information, and I actually have set up a way to, so sometimes you can see me. Look, it works. This is Virology Live. I hope you like the name of the course. I thought that was simple, but tells you what's going on. I am a professor at Columbia University. I have a PhD in virology. I got my PhD here in New York City in 1979 with Peter Palese working on influenza viruses. I then went on to do a postdoc with David Baltimore, not too many years after he won the Nobel Prize for discovering reverse transcriptase. And then I worked in his lab on polio virus. Then I got my position at Columbia University in 1982, where ever since I have been doing research on viruses, teaching virology, writing textbooks, and more recently, of course, podcasting and blogging. And in the last 5 years, of course, my colleague Amy Rosenfeld has played a huge role in the direction of our laboratory. So you can find me at many places online. The email you can reach me at for questions and so forth, Vincent at Microbiome TV. I am on Twitter where I'll announce class sessions mainly. I don't go there to chat. I chat here and on my podcast. I'm on Instagram where I'll show pictures of what's going on in my communication world. YouTube, of course, is where you are now to see this live stream. And there's also a Facebook page where I mainly use social media to post about the communications that I'm doing. So this is the website for Virology Live. It has already the schedule. These are going to be Mondays and Wednesdays, 11 a.m. Eastern. Some days we won't have sessions for various reasons. I have things booked way ahead of time. And so there'll be, I think, 3 days where we don't have sessions. So check the schedule before you come here. There you'll be able to find the schedule. The slides I put up as PDFs, so you can download them, have them. You can do what you want with them. Just don't publish them because most of the figures are owned by ASM Press. There will be links to study questions that you can use to review your understanding. There are readings. There will be a link to the live stream once it's posted. All of these are recorded, of course, and they're stored on YouTube, and we'll put a link to that. And then every week there'll be an online quiz, just 3 questions per lecture, just to test your understanding of things. Now, you don't have to get a textbook, excuse me, for this course. But if you'd like to buy one, this is a great one because it is what the course is based on. It is Principles of Virology. It's in the 5th edition from ASM Press. If you already have it, that's great. For every session, I have a chapter from the textbook that you could read to increase your understanding of what I'm telling you. Now, full disclosure, of course, I am an author of this textbook, and I make money from it. It's about the only thing I do aside from working at Columbia, for which I get income personally. Everything else goes to the communication. So, if you want to support us, that's great. But as I said, you don't need it to do well in this course. And there are going to be ways that you can assess your progress, as I mentioned. Here's the table of contents for the book. And on the left is Volume 1. And you can see there are no chapters on viruses except for one, and that would be HIV type 1 pathogenesis. That's the only virus that gets its own chapter because it's been causing a pandemic since the 80s, unabated. And we have learned so much about it. So much work has been done. And that deserves a chapter in its own. But everything else is principle-based. You can see Chapter 1 is all about molecular biology, how viruses reproduce in cells. Volume 2 is all about viruses and disease. You know, how viruses cause disease, immunology, vaccines, antivirals, evolution, emergence, and so forth. And so we will have sessions in this live stream course that largely mimic the chapters in the book. Not all of them, because I think we have something like 25 sessions here. So I had to leave some out and make some adjustments. But that gives you an idea of what we're doing. Also in the syllabus, you will find links to articles on Virology blog. So I've written Virology blog since 2006. It actually predated the podcast, This Week in Virology. And every week I write something. And I have two other co-authors who write with me. David Tuller writes about CFS, ME-CFS, chronic fatigue syndrome, and more lately, long COVID, which seem to have some similarities with ME-CFS. And then there's Trudy Ray, who writes about virology. I will link to articles that I think are good background during each session. You don't have to read any of this that I give you, but it would enhance your understanding. You know, the more you wrap your head around the subject, the better you're going to be to understand it. And then, of course, This Week in Virology, I will occasionally link to episodes. But, you know, if you have time, I would highly recommend listening, because you'll hear a bunch of experts talk about viruses, people who work on viruses or write about them, some people who do the experiments. We have guests quite often. I don't think there's any downside to listening, so I highly recommend that as well. As far as this series goes, Virology Live, we will have during the lectures some quizzes. So coming up today, you'll see there's a website you need to go to. And you take the quiz there, and then I'll show you the results in real time, and we'll see if you're getting what I'm saying or not. It's a lot of fun. It's Socrative.com, and I'll give you the link in a bit. And we do that in my Columbia course, and it's really instructive. There will also be a weekly quiz separate from the ones we have here on the live stream. The weekly quiz will be based on the previous week's lectures. There'll be 3 questions per lecture. It'll be online. You can take it at your leisure. They'll go up on Thursdays. They'll stay up for about a week, then they'll be taken down, which makes you keep up, basically. So this week it'll go up for this lecture on Thursday. And then I'll put up 2 exams if you want to try them. Look, you don't have to do any of this. You don't have to take the quizzes. You don't have to take the exams. If you want to learn more, I'd suggest you try it at least. So we'll have a midterm and a final, which will be online. You can take them at your leisure and see how you do, see how you're picking up the material. All right. And then, as I said, there'll be quizzes during the sessions. I've already told you about that. And I please ask you to ask questions that are relevant to the lecture. As we take our online quizzes during the lecture, I will pause and take some questions from the chat. I can't take all of them, obviously, because I'd like to limit these sessions to 2 hours for your purposes and mine as well. I have a few other things to do, and I have another 2-hour live stream tonight. I'm not complaining. I'm just saying we got to apportion our time. And I'll pick out questions during the breaks, okay? All right, and that brings us to our let's see if there are any pressing issues here before we go on. Let me just put it on me, and I'll check out the live stream here. Okay, so it looks like everything is good. And we'll get right to the lecture, or the discussion, I should say. We live and prosper in a cloud of viruses. Viruses infect every living thing on the planet. If you look at a living thing, you can assume there are viruses in it. Humans, we regularly eat and breathe billions and billions of virus particles on a daily basis. And we'll talk more about how that happens. I think before the pandemic, many people felt that viruses infected you now and then, and sometimes you got sick. But it turns out that's not correct. Viruses are in us all the time. In fact, we even have genomes as part of our own genetic material, viral genomes. And we're going to explore all of this in this course. One of the principal strategies for viruses to propagate is to make lots and lots of particles. And that it makes sense because they have to bump in randomly to a new host. When you think about it, many other animals don't make a lot of offspring, depending on how tough it is to exist in the world. Rodents make big litters. Humans and elephants, not very big typically, because the offspring are well cared for. But viruses, when thousands and thousands of viruses come out of a cell, they have to find a new cell. So making more of them increases the probability that they're going to find a new cell. So the numbers of viruses on Earth then turns out to be, as a consequence, staggering. For example, bacteriophages, viruses that infect bacteria. You see an example of one here on the cover of Cell here. In the waters of the world, principally the oceans, there are a lot of bacteria. And, of course, there are viruses that infect them, bacteriophages. There are over 10 to the 30th particles in the oceans. That's a huge number. It's bigger than Avogadro's number, right? And it's hard to really appreciate how big it is. So we can do two thought experiments that help. And the first is we know the weight of a bacteriophage. They're about 10 to the minus 15th grams. So if we multiply 10 to the minus 15 by 10 to the 30th, we see that the biomass of these bacteriophages just in the oceans exceeds that of elephants by 1,000-fold. These are things you can't see, 1,000-fold more than elephants. And if you lined up these bacteriophages head to tail, they would go 100 million light years from Earth, which is way beyond the former planet known as Pluto, way beyond the closest galaxy. It's really a long line. That's how many there are. It's really remarkable. Another example for the oceans, whales are commonly infected with small RNA viruses called caliciviruses. We have versions of these that infect us. They're called noroviruses. They can cause illness in whales and other marine mammals. Sometimes they infect us. But the point I want to make here is that whales excrete over 10 to the 13 caliciviruses daily because they excrete it in their feces. And so each whale is pumping out lots of caliciviruses. They add to the number of viruses in the oceans, of course. And they infect other marine mammals. And occasionally humans can be infected by these viruses as well. I think if you got one message out of this whole course, it's that viruses are not just purveyors of bad news. They can do good things. We have some evidence for that, especially in experimental systems that we can manipulate. We don't have a lot of evidence in humans. I'll give you some in a moment. But if we go back to the oceans, you just take a liter of coastal seawater. Next time you're at a coast, take a liter of water. Well, you could just take a teaspoon, actually. I think 2 slides ago I had a teaspoon here. A few million virus particles in a teaspoon of water. But if you wanted to take a liter, there'd be more viruses in that liter of coastal water than people on Earth. I think that's just remarkable. I never get amazed. I never fail to be amazed by that factoid. And so here we have 2 ways of displaying that information in pie charts. On the left, we have by biomass. So we take prokaryotes in yellow, protists in red, and viruses in blue by biomass. And of course, the prokaryotes rule by biomass. They weigh more than anything else. But if you look at particle abundance, so you just count particles, so a bacterium is a particle, a protist is a particle, a virus is a particle. Viruses rule then. 94% of the particles in the ocean are viruses. And this is not something we knew until about 15 years ago, actually. We thought, first of all, that microbial diversity in the ocean was low, and that was wrong. And we didn't know there were so many viruses, and that was wrong as well. And it turns out that all these viruses in the ocean have important functions. They are catalysts for biogeochemical cycling. What does that mean? It means that the viruses kill bacteria. The bacteria break up into large chunks of organic material that sink, and then those are further processed by other bacteria and other organisms to give you organic matter that is recycled. A lot of the carbon, for example, that's recycled out of the ocean comes from viruses killing cells. So they're really important. And we normally in this course have a session on ecology, which explores that more. Unfortunately, we don't have time to do it unless there's great demand, and I push the end of the course one session past it, and then I can put in my lecture on ecology, which is really cool. And it talks about some of the ways that viruses in the environment play big roles. Another number that's very important, there are 10 to the 16 HIV genomes on the planet today. I have to pause because I just saw a question. What's a prokaryote? Okay, a prokaryote are organisms without nuclei, like bacteria, essentially. All right, that's a general name that we give for organisms without nuclei. It is different from a eukaryote, which are organisms with nuclei. Okay, so yeah, I will look at the chat. Protists or protozoans. They're eukaryotes that are typically single-celled organisms. Think of perhaps amoeba. But yes, you could Google a lot of these terms while you're listening as well. All right, 10 to the 16th HIV genomes on the planet today. That is because we know how many people are infected, over 30-some million. We know how many genomes they have typically. It's also a huge number. What does that mean? Well, it means that in that 10 to the 16th genomes, there's already resistance to all antiviral drugs that we own against HIV. There are about over 30 of them. There's already resistance out there, and there's resistance to any we will ever make in the foreseeable future. That's how big a number 10 to the 16th is. And why this is, we will talk about later in this course. Now, each of us are highly infected with multiple viruses. We have at least a dozen herpes viruses. These are all acronyms for different herpes viruses. They're more than just herpes simplex 1 and 2, which many of you have likely heard about. We have varicella zoster virus. We have human cytomegalovirus, Epstein-Barr virus. And then the more recently discovered ones were just given numbers, human herpes virus 6, 7, and 8. And we'll explore how some of these work later on. But all of you, nearly all of you, are infected with all of these viruses, because once you get them, and you get them typically in your first decade of life. For Epstein-Barr, sometimes you don't get it till you go to college. You get them all eventually from people who are close to you. They're typically spread by close contact, saliva, for example, kissing, and you keep them forever. Once infected, it's for life. And I love to say, at this point, unlike love, herpes is forever. And those of you who've taken my course, remember that. It's true. They all stay with you forever. You can't get rid of them, despite what people are saying. They say, oh, I was cured by Dr. So-and-so of my herpes. No, you're not cured. Maybe in the future we can cure you, but you're infected for life. So at least all of these herpes viruses, but we have many, many more viruses. I'm sure all of you know that we have a microbiome. Every part of our body has a unique microbiome, whether it's the mouth, the respiratory tract, the stomach, the intestine, the skin, the urogenital tract. Every one has a unique set of microorganisms that contributes to our well-being. They're good. They're beneficial. Microbiome's gotten a lot of study because it's actually relatively easy to determine microbiomes. You just need to sequence the ribosomal RNA of the bacteria. It's different enough from our ribosomal RNA, and it can give you the species of the bacteria, or at least at a higher taxonomic level. And it's easier than of figuring out a virome. We have viromes, but we have to sequence the whole genome to sort it out. So we each have a virome, and this picture shows you the DNA number of DNA viruses and RNA viruses that have been found by doing sequence analysis of different parts of the human body. You can see the nervous system, respiratory tract, urogenital, digestive, blood, skin, hair, nail, all of these are in healthy people. Look at the numbers. So all of us have a lot of different viruses in us that apparently are not causing problems. And so this is called the virome, and this is why, in part, I firmly believe, and so do many others, that these are doing good. How they're doing good, we don't know. It's hard to experiment, right? We can't really experiment on people, but we can do it on animals, and I'll tell you some examples of that in a moment. Now, the virome can easily be determined for many different kinds of animals. You just take samples and sequence them. We're learning now that the environmental virome is getting easier to sample. It's easy to take seawater, right, and just sequence it and see what virus is in it. It's easy to sample small living things, but what about whales? Can we sequence their virome? And the answer is partially. This is a movie I'm going to show you in a moment that shows how a drone has been adapted to sample the whale breath virome. So here is a drone which is being run by scientists on a boat. This is actually off the coast of Australia. So those of you listening from down under, this was done off the Australian waters. And they followed this pair of whales here. You can see they're breathing right there. Their plumes are up. And the scientists this is a big petri dish here, and it can be opened automatically when the whales are breathing. Let me play this movie. Hopefully it works. There you go. There you go. So the whale there blew, and the plate open, and they captured the breath. We'll play it again because that's going by pretty quickly. Breath, open up, capture the drone is steered through the breath, right? They bring it back to the lab, they extract nucleic acid, they sequence it, and they find that there are tons of viruses in whale breath. Now, how they relate to what's actually in the whale is hard to know, right? Because some of them can be just picked up in the water, the ocean water, as the breath comes out and so forth. But look, this gives you the ability to sample wildlife that you can't normally interact with. So there are a lot of possibilities for that. As I mentioned earlier, we have DNAs as part of our own genome, viral DNAs. Right? These are viruses that infected us years ago and stay with us. This is a pie chart of the human genome, 3.2 billion bases. It's remarkably hardly, I've lost my pointer here, it's hardly, excuse me, hardly any coding sequence. That's the red part. 1.5% of our 3.2 billion base genome codes for proteins. The rest, some of the rest does, but they're typically interrupted. So you can see lots of introns, intervening sequences. And then on the upper part of this, let me see if I can recover my pointer. Yeah, there we go. The upper part of this, LTR retrotransposons, about 8%. Those are retroviruses that infected us many, many thousands of years ago. The DNA has remained because it integrated into the germline, of course, to be in every human. Every human has these LTR retrotransposons, and exactly what that means we'll talk about later. Every human has these sequences. In humans, we don't make infectious retroviruses from them, but other animals, like mice, have LTR retrotransposons, and they can make infectious viruses. So it's a way to ensure transmission of the virus to another host. And we'll explore these LTR retrotransposons. We're going to talk about how some of the genes are actually used by us. So one of these genes is used to make placentas. We'll talk about that. Now, of course, a major driver of interest in viruses is that they cause human disease. And here is a chart showing the causes of 2017 global deaths from this website here, IHME Global Burden of Disease from Our World in Data. Great website. And we have the biggest killers, of course, cardiovascular diseases, followed by cancers. Then we have respiratory diseases, many of which, of course, are viral. We have in particular lower respiratory infections. We have diarrheal diseases. So the big red arrows are ones where viruses make a big contribution. HIV/AIDS on its own, hepatitis down there. And so many viruses cause significant human deaths. And of course, we have COVID here from, as of last night, 4.7 million deaths globally, which is surely an underestimate. It's probably over 6 million. So it's, I suspect it's going to move into the second position for a while, right? Which is really stunning. And this drives interest in working on viruses. But there are many, many other reasons to be interested in viruses. Here's the latest Johns Hopkins panel on the COVID pandemic. I picked this up last night. It's a screen capture. Total cases that have been detected, of course. We don't actually know the number of cases. It's probably at least a 5-fold underestimate. The total numbers of deaths. And you can see the distribution. Early, we followed this, as many people did early in the pandemic. In fact, I was teaching a virology course at Columbia in the spring of 2020, and we followed this. And for many, many days, there were just dots multiplying in China and nowhere else, and then suddenly started to multiply elsewhere. It really was quite eerie. I hope someone has made a time lapse of that proceeding. I'm sure they have. Anyway, this drives interest, of course. But despite these viruses causing disease, the vast majority of them don't harm us or any of their hosts. And why is that? Why don't most of the viruses that infect people harm them? Well, in fact, most of them just pass through us. We eat many viruses on a regular basis with our foods. For example, there was a study done a while ago of cabbage from 5 different supermarkets in Washington, D.C. They bought cabbage, they brought it into the lab and looked for viruses, and they found that each serving of cabbage, making coleslaw or whatever else, would have up to 10 to the 8th particles of an insect virus. It's called a baculovirus. It's shown here in this picture, very long DNA viruses, and in this case, they infect the cabbage looper, which is an insect, a caterpillar that crawls over cabbage and deposits viruses. And you can't remove them from the cabbage. They're adhered pretty tightly, and you end up eating them. They pass through you because they can't infect your cells. And in general, viruses from plants and insects and other species distantly related to us don't infect us because the cells are very different. And you can now, of course, look in human feces for viral sequences. You can actually send your feces to a company, and they will tell you what your fecal virome is. And the most, as Myra Breitbart mentioned on Twiv a few weeks ago, most of these viruses that you find in human feces are similar to plant viruses, and the most abundant one is pepper mild mottle virus, which causes a mottling disease of peppers. As you can see here, there are 10 to the 9th virus particles per gram of dry feces. So many people eat peppers, the virus is in it, and it passes through you. All right, so this is common. Many viruses we ingest on a daily basis, and they pass through us. Let me give you some evidence that some viruses are actually beneficial. And here are 2 from systems that we can experimentally manipulate. So the first on the left is a grass called Dicanthelium lanuginosum. This is a grass that grows around hot springs. If you've ever been to Yellowstone National Park in the U.S., you see the hot springs. Around the edges, there are grasses growing. They can handle the high temperatures, and this is one of them. The ability of this grass to grow at high temperatures is due to a fungus inside of it, which in turn is infected by a virus. So this is the fungus here, Curvularia, that grows in the plant itself, and the fungus in turn is infected with a virus. And both are needed for growth at high temperature. If you take out the fungus and the virus, the plant cannot grow at high temperature. If you leave the fungus only but not the virus, the plant will not grow at high temperature. And you can do those kinds of experiments in the laboratory. So we can tell that the virus host system here, the virus-fungus system, is beneficial. And that's the kind of thing we can't do in people. On the right is another example of a beneficial virus. This one is beneficial for these parasitoid wasps that inject their eggs into an insect larva, and then the eggs develop into larvae and new baby wasps, and they eat the caterpillar from the inside out and then burst out at one point. So this has been happening for thousands of years. You know, alien didn't invent anything. It's been happening with parasitoid wasps for a long time. So where does the virus come in? Well, it turns out that when the wasp injects the eggs, along with the egg come viruses. These are DNA viruses called Polyoma viruses that are encoded in the wasp genome. So the wasp genome encodes for these viruses. They do not reproduce in the wasp, actually. They are just assembled in the ova of the wasp. They're delivered. The virus particles are made and delivered along with the egg. So what do? They infect the caterpillar, actually, and they immunosuppress it so that it will not reject the wasp egg. Right? That is really amazing. There are many, many such amazing stories in the virus worlds of plants and insects, because those are systems that we can manipulate to study. So this is a virus beneficial, obviously, for the wasp, but not for the caterpillar. What about mammals? So I think this is the best example so far. This is an experiment done in mice, so you can easily study this kind of virus-host interaction in the laboratory. So we have, this is the intestinal tract of mice here, a section, and you can see the villi. And we have three different kinds of situations here. We have conventional mice on the right, CONV, where these mice are grown normally, so they have their complete microbiome in their gut. The commensal bacteria refers to the gut microbiome. And everything is normal. The villi look good. The immune system is developed. These brown cells in the lower two sections, those are lymphocytes that populate the gut, and they're normal numbers there. All right, so now if you grow mice under germ-free conditions where the gut microbiome is absent, now you see that the intestinal villi are defective, they're malformed, and there are no lymphocytes, highly reduced numbers of lymphocytes. So apparently the gut microbiome is important for development of the gut and development of the immune system. Remarkably, if you now take these germ-free mice and infect them with a murine norovirus, MNV, it partially restores the morphology, and now you have lymphocytes coming back into these animals. So a virus, an enteric virus, can apparently replace the beneficial function of commensal bacteria. I think this is a really good experiment done by Ken Cadwell's group here at NYU, which is just across town from me. And the beauty of the incubator is that I can get these people in here one day and talk to them about this work, and you can listen. So basically, the other reason we don't get sick, besides viruses passing through us or being beneficial, we have an amazing immune system, which we will cover briefly in this course. It's not an immunology course, but we have an amazing immune system that will take care of most virus infections. And in fact, in these commensal viruses that are in us, we think the immune system keeps them in check. And if your immune system is down, which can happen if you're immunosuppressed, if you're on immunosuppressive drugs, if you have a virus infection that's immunosuppressive like measles virus or HIV, in those cases, then your immune system is suppressed and those viruses in you get out of control. So, for example, people who receive organ transplants receive immunosuppressive therapy so they don't reject the organ. Those individuals have reactivation of many viruses in them, like cytomegalovirus, and they can cause huge problems. Now, as I hinted with other animals, not all human viruses make you sick either. We have lots of viruses in us that don't make us sick all the time, and one of them is this Polyoma virus, a small double-stranded DNA virus that will feature highly in this course. as we go through basic molecular biology. This infects pretty much everyone in your first few years of life, and you typically get it from your family. And families in different parts of the world have different polyoma viruses that they transmit that can be distinguished by the antibodies that you make. So we can take blood from people and look at the kinds of polyoma viruses they have. And in this slide, I show you that we can actually track human migration out of Africa by what kinds of polyoma viruses have infected people. You can see the origins of Homo sapiens in Africa, moving out into Europe, Asia, Australasia, and eventually the Americas. We can track this by the kind of polyoma viruses people have. And even more amazing, it matches the movement out of Africa, but the dotted line is the movement tracked by the genome of humans. And so it's actually a little bit more detailed. Viruses also shape host populations and vice versa. So viruses can limit the way host populations emerge. They can enhance them. And if a host goes away, the virus goes away too. So it's a two-way street. And here's a great example of that. This is a photograph from space of a planktonic bloom in the ocean. And that white material is mostly Emiliania huxleyi, a protist that grows to high numbers, enough numbers so you can photograph it from space. And there are, of course, viruses that infect E. hux, as it's called, and it destroys the blooms. So when the blooms grow, which are typically stimulated by sunlight, the virus activity increases and the bloom collapses. So huge regulators of ecosystems. You know, these are just some examples of why I think viruses are amazing. And what I want you to understand also is that virology is an integrative science. You don't just learn one thing. If you want to learn, chemistry, you learn chemical reactions and elements and so forth, how they combine and break down. I mean, not to say it's not complicated, but you learn chemistry. But for virology, you need to learn everything. You need to learn chemistry, organic chemistry, biochemistry, cell biology, physiology, molecular biology, genetics. You have to learn sociology and psychology and epidemiology to understand how viruses spread. So it's an integrative science that makes you understand many, many things at multiple levels. Now, this course is designed to help you see this big picture. I want you to think about virology as an integrative discipline, not influenza and herpes and SARS-CoV-2. You don't learn viruses that way. I want you to think of the big picture. We can certainly address specific questions, no problem. But the main goal is to give you a big picture, and you're going to learn about fundamentals. You're going to know more virology than most people around, and this is stuff that people don't know, and as a consequence, they're frightened. And you're going to learn it, and that is what my goal is. Someone is saying, what are these viruses? So the virus on the left is poliovirus attached to its cell receptor. And on the right, it is a giant virus from the oceans. That is a virus that infects a protist called Cafeteria rowenbergensis. Huge, huge virus. We're going to talk about that later. Now, one of my big beefs, if you followed me at all, is the way virology and science in general is portrayed in the mass media. And pre-pandemic, I used to collect headlines to show in this slide about from influenza pandemics. But now, of course, these are from the COVID pandemic. And these are some headlines that illustrate my disappointment in the way mutation in particular is portrayed in the media. So here, these are all from quite early on in the outbreak, except for this one down here. Don't go to Wuhan. Don't leave Wuhan. Coronavirus could mutate. China warns coronavirus could mutate and spread. And here we have the national health minister. The virus is transmitted through the respiratory tract. There's the possibility of viral mutation. This drives me nuts because, as you will learn, and many of you already know, viruses always mutate. The question is, do the mutations matter? And the subtleties, there's always nuance. They're often missed in the press. And much of what is reported is simply wrong and misleading. And I'm here to try and steer you through that. And many people are grateful that we can do this. Okay, so let's try this quiz here. Now, what you do is you go to socrative.com/login/student. You can just go to socrative.com and click on the student login. It'll ask you for a room number, and that room number is virus. And then you're going to be presented with a quiz, which is right here, and you can pick your answer. And we're going to, after a few minutes, I'll stop and see how you did, because I can show you the bar graphs of the answers. So here the question is, which statement is true? All viruses make us sick and can be lethal. Our immune system can manage most viral infections. Humans are usually infected with one virus at a time. The press is usually correct in their virology reporting, or our immune system cannot handle most viral infections. Okay, so check that out. Hopefully you can get there. I'm going to go to the website and see this out, Socrative. Okay. I can see some of you have already answered. That's good. And I am going to put that up now. There's the Socrative interface from my view. So you can see the question, and I see how many students are here and how many have answered so far. Right. And then when we've reached, let's see, how many people do we have here? We have 430 people. Wow. That's certainly more than I taught in any class. 430 people. So I don't expect all of you to come here, but we'll get to some number where the rate of it accelerating. So in the meantime, let's see if we have some questions. So I have a journalist here who says, I understand you, Vincent. No, I don't mean to indict all journalists, right? I can actually, let's see, can bring up these questions here on the chat, and I've already lost it. So, but for example, I can bring up your chat comments here and answer them individually. I'm sorry if you're having trouble with the quiz. Obviously, a lot of people have. 90 students have done it, and there are hundreds and hundreds of questions. Oh my gosh. There was a good question. Well, now that I'm looking here, I can thank Kent for his super sticker. You have ways of supporting us if you'd like. You don't have to, but we really appreciate it. Videographer says, I eat viruses for breakfast. It's absolutely true. You do. How are we doing on the quiz? 110. So I'm going to stop there because it's not going up anymore. Oh, here are 2 questions I wanted to answer. Let's go back to all these buttons in front of me. There we go. It's working. Are there viruses of viruses? So there are viruses that have to infect a virus-infected cell, for sure. Okay? There are no viruses that infect another virus particle. That can't happen because the virus needs to infect the cell. It can't reproduce on its own. And we will actually talk about that later on in this course. All right, that was one that I saw. What was the other one? Viruses of virus. Okay, well, what percent of our genome is viral, roughly? So about 8% or so is for humans is viral. And, we have some moderators here who are highly familiar with viruses. In fact, one of them, Vanity Nutrition, took my virology course at Columbia, so she's also qualified to answer your question, as are others. And since there's so many, we'll have to depend on that, right? All right, we have 140 students. Let's stop and see how we did. Okay, so now I click this button that says Show Results, and so this is great. 95% of you got the right answer. Our immune system can manage most viral infections. They do manage most of the infections, and only when the immune system is down do we have a problem. A few of you said humans are usually infected with one virus at a time. I remember I showed you that we have multiple herpes viruses most of our lives and many other viruses. The press is usually correct, and probably there's a journalist or two in there. They're not usually. It depends what kind of press. Mass media, big headline press is just a headline saying the virus is mutating is simply wrong. And that happens over and over and over again. And our immune system cannot handle most viral infections. They actually can. They can handle most virus infections. And so that's B. Most of you got that. So this is how this will work. And I will try and pull out some questions along the way. I can't pull them out all, obviously, because there's so many good ones. Let's see. Just a few more. By the way, I wonder, see, you can see me next to the slide. Do you need to see me because I make a lot of gestures, or would you prefer to just have the slide? Why don't you let me know in the chat? What came first, viral or cellular life? Oh, we're going to talk about that in evolution. Yeah, there's only one question so far, folks. You're going to get another one in 15 minutes or so. Okay? You'll get used to it. Don't worry. So let's continue with some slide. This is fine. You like seeing me? Okay, fine. If you don't, put it in the chat. All right. Now, after almost an hour, let's define a virus. Full screen is better. Full screen or clearer text. Okay, we'll go full screen then. Let's do that. And I'll do the me for the questions. What's a virus? As I said, folks, this is an experiment. We're going to have things in the beginning that need to be changed. I totally understand that. What's a virus? Here's my definition. An infectious obligate intracellular parasite comprising genetic material, DNA or RNA. Sometimes it has a protein coat, often surrounded by a protein coat, and sometimes a membrane. So let's break this down. So an infectious, you understand, is something that can move from host to host. Obligate means it has to, and intracellular means it has to get into a cell. So obligate intracellular means the virus has to get into a cell to reproduce. No exception. Every virus, in order to make more viruses, has to get inside a cell, and that's why we call them obligate intracellular. And then the parasite part is because they take something from the host. So parasites take something from the host. They don't necessarily kill them. You can steal energy, and that's enough to be called a parasite. And, we all know human parasites, right? You get the idea. All right, so that's the first part, comprising genetic material. So all viruses have genetic material. It can be DNA or RNA. And RNA viruses are unique on the planet because they're the only organisms with an RNA genome. Everything else is DNA-based. But they are relics of a part of history on Earth called the RNA world, when everything was RNA. And we're going to talk about that when we talk about evolution. So RNA viruses are relics of a very, very ancient time in history of Earth. And then, surrounded by a protein coat, pretty much all the viruses that we talk about are surrounded by a protein coat. And these schematics are all schematic drawings of different viruses. And here down at the bottom is our coronavirus, where the RNA genome is shown as a coil, and then it's got protein on it, and then there's also a membrane. So sometimes viruses have membranes. So on the left of the coronavirus is adenovirus, which is a pure protein shell. And inside it is the viral DNA genome. So there's one that doesn't have a membrane. So protein code is obligatory, but the membrane is not. Many viruses do not have membranes. There's a polio virus without a membrane and many others. And we'll talk about structures in a bit next week, and you'll see how these are built. So that's the definition of a virus. And when you study viruses, you learn about the host. Because viruses are obligate molecular parasites, right? They depend on the host. They need many, many different systems from the host, many of which we'll go through as we chat some more. So if you figure out a way a virus does something, it tells you something about the host as well as the virus, whether the host is a mosquito or... Here, this, by the way, to the left of the human is a protist called Cafeteria rowenbergensis. You can see it's a single cell, has a nucleus, has a flagellum. These are very, very numerous in the oceans. And they're probably the single most numerous protist in the ocean, and there are viruses that infect them. That's that big virus that someone asked about on that pretty picture a few slides ago. So you learn about all these hosts by studying viruses, essentially. Okay, now, are viruses alive? This is what many people would like to know. Many people have opinions. I thought about this for many years. And we put a poll up on my blog some time ago. I don't think it's up anymore. You know, the polling software kept changing, and at some point I had to pay for it, so I stopped. But after a few thousand votes, look, yes and no were pretty much evenly divided. Something in between got quite a few votes, an equal number of votes. And then people said, I don't know, a few people. So are they alive? I have an opinion, and it's many virologists differ, but let me tell you what I think, and you can decide whether it makes sense. I think a virus is an organism with two phases. So when you say virus, you have to be really specific about what you mean. You have to, in my case, say it's an organism with two phases. Some people, when they think about a virus, they just think of the infectious particle. So here on the left is the infectious particle. So that's one of the words I use to describe a virus, infectious particle. It's also called a virion. So a virion is a very specific word meaning the infectious particle. As you will see in a bit, not all viruses as they come out of cells are infectious. Some of them are broken. They're not infectious. So we call them virus particles, but the infectious particle is called a virion. So that's one of the phases of virus. This in no way is alive. It's a piece of nucleic acid wrapped in protein at its simplest. It can't do anything unless it gets into a cell, and that's shown on the right here. And then we have an infected cell, a virus-infected cell. This is the reproduction cycle of a polyoma virus. That happens to be what this virus is. The polyoma viruses that infect most of us. Virus binds to receptors, gets into the cell, directs the synthesis of new viruses. By the way, if you're hearing background noise here, it's because I haven't completed my soundproofing yet. It will happen eventually, so I apologize if you're hearing street noise, etc. I'm hearing music outside right now. So this is the infected cell, and it's taken over by the virus, right? So it is alive. At some point, it may no longer be. The virus infection may kill it. But for a number of hours, it is living. It is certainly part of the organism. It's a virus-infected cell, so it's living. And I have no problem with saying it's alive because there the virus can reproduce and evolve and so forth. So that's my interpretation. When you think of virus, think of an organism with two phases, and then you can easily say whether, yes, a virus is living because it's the infected cell part that's alive and the particle is not. Now, the other thing I want to warn you all about is that you can't use anthropomorphic analyses to talk about viruses. This happens all the time because it's very easy to do, and that's fine. You know, the press does it. So even scientists do it. I try not to. viruses are not sentient. They're not animate. They don't think. They don't employ. They don't insure. They don't do anything that humans do. They don't achieve their goals in a human-centered manner. They're passive agents. They come out of a cell. They bump around until they find a new one. They're taken up passively into the cell. They have no influence on what happens whatsoever. So it's not correct to give them anthropomorphic properties, because what you're doing is you're applying human values to them, and you have no idea whether that's correct or not. So I would avoid it. When we wrote our textbook, we actually had to go through many times to take out all the anthropomorphisms. And you will all do this. It's fine. I just want to warn you why it's not appropriate, because you apply human values to viruses, which, as I say, may not be correct. Part of the definition of a virus is that they're very small. In fact, let me back up. A part of the definition that we used to have is that they're very small. That's gone now, because the first viruses discovered were, in fact, very small. So here's a figure that gives you an idea of that. Here's an E. coli, for example, at 100,000 X, and attaching to it is a bacteriophage. So you can get a sense of the size of the virus, the bacteriophage, with respect to E. coli. Here is tobacco mosaic virus, this rod-shaped virus, which we'll encounter in a moment. First virus discovered. And there in panel D is HIV. So you can get an idea of their sizes relative to the host. Now, this panel here has a collection of viruses and molecules. It's amplified a millionfold on the right. And here we have a ribosome in G. We have a tRNA in E. We have an antibody molecule in F. And here at the bottom is a poliovirus. So you can see it gets down to very, very small sizes. These are microtubule and actin filaments and a variety of enzymes from cells. So they're quite small, and in fact can even drill down deeper into size. This is a chart showing you a range of sizes of biological materials. So we're going here from plant and animal cells that you can easily see in a light microscope, of course, bacteria, which you can easily see in a light microscope. And then we have viruses, which the biggest viruses, the giant viruses, you can actually see in a light microscope, but you typically need an electron microscope to see them. And then ribosomes, as you saw in the previous slide, can be the size of some viruses. Proteins then are even smaller. You can see some proteins by cryo-EM or X-ray crystallography. And then you can go all the way down to atoms, but you need techniques like cryo-EM, X-ray crystallography, and nuclear magnetic resonance, things that we will talk about subsequently. So the viruses fall into this intermediate size group. And you can see another nice photo of a cell here, a drawn image with its nucleus, mitochondria, endoplasmic reticulum, Golgi. And here on the outside of the plasma membrane are some virus particles. And we can blow that up, and you can see we have a herpes virus here, which is about 200 nanometers. And the poliovirus, about 10-fold smaller, 30 nanometers. So big range in virus sizes. If you're curious about how many viruses fit on the head of a pin, well, of course, it depends on the virus. But if it's a rhinovirus, one of the common cold viruses, you could put about 500 million of them. Here's a pin, and it's about 2 millimeters in diameter, the top of the pin, which is about 2,000 microns. So we use dimensions like microns or angstroms. We'll get more into those sorts of dimensions later. But in this little area here, so there's a dust mite on the top of the pin. And in this very small area, which is blown up on the right, we have some red blood cells, some lymphocytes, some yeast, some bacteria, green and yellow. And then there, you can barely see it. It's Ebola virus, which happens to be quite long. So the viruses are quite small relative to the head of a pin. And so when you sneeze, the droplets that you expel have many, many thousands of viruses in them. Now, as I said, we used to think that viruses are very small, but about 15 years ago, we discovered viruses, which broke all of our ideas about how small viruses were. And here's a lovely cover from the American Scientist from 2011. Here's rhinovirus and HIV, slightly bigger. And here's Mimi virus, much, much bigger than any known virus. In fact, the biggest known virus when Mimi virus was discovered was this virus of algae. And you can see here in the same cell are two Mimi viruses looking at you, and they are substantially bigger. So we'll talk about giant viruses later on in this course, but no longer do we say viruses are small. In fact, some of them are quite big, and you can see them in the light microscope. Here's a Pandora virus, which was discovered a few years after Mimi viruses. These are very large DNA-containing viruses. There's a photograph, an electron micrograph of them there, and a drawing there. They have a membrane around them. They have a DNA genome, and there's a pore at one end that we think opens up to let the DNA out. And these are about 1 to 1.5 microns in length. You can see them. This is a light micrograph where you can't see most viruses, but you can see them here. It's really, really remarkable. And here's a graph on the left of virus size, genome size versus virion size. And so here are the giant viruses. So vaccinia is considered a big virus, but you can see it's dwarfed by Mimi viruses. Here's Pandora virus here. The biggest particle size are the pithoviruses, which are in excess of 1.5 microns or 1,400 nanometers in length. But the biggest genome size is the Pandora virus, which is 2.5 million base pairs of DNA. It's absolutely huge for a virus, bigger than some bacterial genomes. Really remarkable. The other key part of the virus definition is that the viruses replicate by assembly of components that are made in cells to form virus particles. And that's very different from bacteria and from cells, which divide. So here on the bottom is the growth curve of a bacterium. You can inoculate a broth with bacterium. They will start to divide very quickly, and they will divide by what we call binary fission, which simply means one bacterium divides into 2, and those 2 then divide into 4 and 8 and so forth. Binary fission. 1 becomes 2. Viruses don't do that. They don't simply split and make more viruses. The virus needs to get into a cell, make the parts for a new virus, and then get out of the cell somehow. So when you infect the cell, here is a similar growth curve as for the bacteria, except we're looking at time and the number of new particles made, virus particles. When you infect a cell, there's always this delay in the production of new virus particles. We call it the eclipse, when you can't detect any viral infectivity in the cell. And that's because the parts are being made. And finally, when they're assembled, then you see new virus particles made. And that is a key difference between viruses and bacteria. All right, let's take another quiz here, and I go to Socrative and go to the next question. Okay, number 2. So let's put that up. Which of the following is true concerning bacterial versus viral replication? Viruses must assemble using preformed components. Bacteria do not replicate via binary fission as viruses do. Bacteria must assemble using preformed components. Viruses do not have an eclipse period, and viruses replicate by binary fission. Which is true? It's always the best answer. So I see there are 162 people logged in, which is great. And we'll let that incubate, and let's go to some questions here. Did viruses as we know today come before prokaryotes? Well, some people think they did, and it's hard to define what they were then. We know that in the RNA world they were certainly self-replicating nucleic acids like RNAs, and those were probably precursors to viruses. And then when cells arose, prokaryotes, the oldest cells that we know of, the viruses went into them. We're going to talk about that more when we talk about evolution, for sure. But yes, we think they were there beforehand. Could humans live without viruses? I think so. I think we could. I don't know what life would be like. We may be able to figure out how to supplement whatever it is that they give us. I think you would not be so great. You probably couldn't live without your microbiome, right? I'm not sure if it would be fatal without a Virum. We just don't know. That's a great question. What viruses do not have a protein coat? They all do. For a while, that slide, you're pointing out the little often have a protein coat. For a while, I thought viroids were viruses, right? These are naked RNA molecules that can move from host to host and reproduce. But Eugene Koonin convinced me that they're not viruses. They're a different entity. We'll talk about those later on. So I have to fix that slide to say all viruses have a protein coat. That's absolutely right. Absolutely right. It is very interesting. Is there a limit on the size of viruses? I don't know. That's a good question. We keep discovering bigger and bigger ones. I'm sure there's some limit, but I'm not sure we have encountered it yet. We thought there were limits on RNA, and we exceeded those. So I think there are a lot of viruses out there to be discovered that we haven't seen yet. Have any prehistoric viruses been imaged? The oldest, not very old viruses have been imaged. Now, we've recovered some 30,000-year-old viruses from permafrost, Siberian permafrost, but we haven't imaged them. No, it's not prehistoric, I guess. What's the smallest viral genome? Oh, we're going to talk about that next time, so save it for next time. It's a good question. Let's see what we have for our Socrative now. 134. That's good enough. So let's see what we did. 87% of you got the right answer, which is A. Most viruses must assemble using preformed components. That's the correct answer. Bacteria do not replicate via binary fission. That's wrong. They do replicate via binary fission. Bacteria don't assemble using preformed components. They do binary fission. The viruses are the ones that assemble. The viruses do not have an eclipse. They do. I showed you that in the slide. And viruses do not replicate by binary fission. All right, let's go back to slides. Now, since some people like seeing me, we'll alternate. How old are viruses? Well, we can now do evolutionary analyses, which we'll talk more about later, by genome sequencing of not just the virus, but also of the host. And that allows us to look at when certain viruses emerged in certain hosts. We'll talk more about that later. But the oldest one that has been pinpointed by that kind of analysis is a marine, which means in the ocean, not murine, which would be mouse, a marine origin of some retroviruses over 450 million years ago. Now, we don't actually have the genome. We just have a molecular clock that tells us that these were likely circulating then. So the Ordovician period is here when we had our first land plants. But remember, life went into the oceans and evolved there, then came back onto land. And it may be that retroviruses emerged in marine life and then came back on the planet, came back onto land with them. In fact, we did a paper on TWIV not too long ago, which confirmed that by looking at viruses of cetaceans by similar analyses, that they clearly emerged in the oceans and then went back onto land. And in fact, some of them came from land and went into the oceans and then went back. So this is the earliest for retrovirus. But maybe they originated before cells, as I said in response to a question. I'm sorry, I keep forgetting to remove the question. I'm really sorry about that. I wish it would do it automatically, right, after a certain amount of time. Viruses probably originated in the precellular world as self-replicating molecules. And we'll talk more about that later on. Now, we can see references to viral diseases in recorded history. There's a big gap, obviously, but here we have two that are particularly interesting. Here is a flask, piece of pottery from 700 BC, and on it is inscribed in Greek, here this firebrand rabid Hector leads the charge. It's actually from the Iliad. And rabid, of course, is a disease caused by a virus. Of course, back then they didn't know anything about viruses, but it shows you that viral diseases, human viral diseases, were clearly present. And we know this from many, many historical references, smallpox and other viral diseases. Here's one reference to polio from 1580 to 1350 BC, an Egyptian carving showing a priest with a leg called drop foot, which is typical of polio. When the limbs, muscles become paralyzed, you can't hold your foot up any longer. So when you lift your leg, the foot drops. It's called drop foot. It's typical of what we call flaccid paralysis, where the muscles cannot move. And of course, since you can't move them, they lose muscle mass as well. So you can see that the leg is atrophied. So this probably is polio, ancient polio. If you've read the Iliad, maybe you remember rabid. In the 11th century, we have records of, in China, a process called variolation to prevent smallpox. Now, smallpox, we know, is a viral disease. But back in the 11th century, what was observed in China was that smallpox infected a lot of people, but if you happened to survive, you didn't get it twice. They didn't know what the story was, but they said, okay, maybe if we give it to people, then we can prevent them from getting sick. So this was called variolation, where they would take the pustules from a smallpox victim and grind them up. And here you see they're being blown into someone's nose. So an old form of vaccination, not FDA approved. And then those pustules have virus in them. We know that today. They didn't. Maybe a third of these people would die. They would develop smallpox and die. But the rest would be infected, develop immunity. So they were vaccinated. Fast forward to the 1700s, Lady Montagu, who was the wife of the British ambassador to Turkey, they're living in Turkey, and they see this practice happening, this variolation. And she said, oh, this is quite interesting, because smallpox is a big problem in the UK in these years. So she brings it back, and it becomes a more widely accepted practice. And remember, all this is done without knowing what's causing the disease. The only thing we knew is that the survivors were protected. And then finally, of course, in the 1790s, Edward Jenner does experiments in England to establish vaccination as a process. Still, and we will talk about that in the vaccines lecture, but still without any knowledge of the agent. All they know is that if you do this, you protect people. And Jenner, of course, inoculated a child with cowpox pustules and protected. He actually challenged the boy with pustules from a smallpox victim, and he was protected. But now we have to talk about the concept of microorganisms in some detail. This is very important in the history of understanding what is a virus. So this depends on three investigators. Leeuwenhoek, who made the first microscopes, right? He was a lens maker who made microscopes. He was curious about what was invisible to the eye. Now remember, we thought in the world everything we saw was all there was. We didn't think there were microorganisms, but Leeuwenhoek saw small things in water and body fluids, and that was the first idea that there were microbes in our world. Pasteur, who came around later, said these microbes, these bacteria, can do interesting things. They can make cheese and bread and wine. They're actually useful. And of course, Koch then said, well, some of these bacteria can make you sick. And he came up with the Koch's postulates, of course, and showed that a variety of bacteria can, in fact, cause disease. So this is the foundation for our current understanding of microbes. And in fact, I neglect to point out here this swan-necked flask of Pasteur. He showed that if you boiled some broth in this flask, it would never grow bacteria unless you broke the swan neck, because the bacteria from the air can't get in. There were some people who thought that life spontaneously generated, and this really put an end to that. As early as 1728, the name virus, or the word virus, was used to describe any agent that causes infectious disease. So by this point, we understand the concept of infectious disease, that there are maladies of humans and other animals that go from host to host, but we don't know what they are. So they were called viruses, actually. And virus comes from the Latin word meaning poison. So these were thought to be liquids, actually. Whatever was going from person to person was some kind of a liquid. And Pasteur later said every virus is a microbe. So he said, he's talking about bacteria. He doesn't know anything about the viruses we think about today. He is thinking that all these bacteria, we should basically, they're what causing the infectious diseases that we have referred to as viruses. So that's what virus was used to indicate up until later, as we'll see. And the key experiment that changed that was the development by this individual, Chamberlain, who worked with Pasteur, of a filter called the Chamberlain filter. Water in Paris in the 1800s wasn't very good, and especially for his experiments in the lab, he wanted to clean it up. And so he developed this porcelain filter to remove bacteria from drinking water and from lab water. And it was basically an apparatus like this. The porcelain filter was on the top, with quite small pores, and you could filter liquids through it using a vacuum suction. And then what got through the filter is the bacteria are removed, what we would call sterile, and this cleaned up the water. And Pasteur started working on rabies at one point, and he would grind up the brains of animals with rabies, and he put them through this filter, and the agent would go through it. So he said, it's a small bacterium. It's too bad he didn't figure out viruses. But a number of years later, people were working in Europe on tobacco mosaic disease. So here, this was a disease of tobacco, which by then was an economically very important crop. And the diseased leaves, which look like this, couldn't be sold for tobacco. So many labs were working on trying to figure out what was going on. And what they did is they would grind up the leaves and filter them. And they found that the agent went through the filter. And this was done independently by two people, Ivan Oski in 1892 and Bayerink in 1898. He called it contagium vivum fluidum. He thought it was a liquid. And so here we have the agent of this disease passing through the filter that retained bacteria. Now, today you can buy these filters. We buy them for our lab. You can buy them in 0.2 or 0.4 micron pore sizes to retain bacteria, and depending on your virus, they may go through it. So you need a 0.4, but that's not going to keep the biggest viruses out. It's not going to allow, that's not going to allow the biggest viruses to pass through. All right, so these two figured out that the agent of tobacco mosaic disease passed through the filter. But they still thought they were liquids. In 1898, two people showed the agent of foot and mouth disease, a virus of cattle, gives them lesions, which again prevents them from being useful, is filterable. And then this work, combined with that on tobacco mosaic virus, showed that not only does it pass through a filter, but the agent will not reproduce in broth. So if you take the tobacco mosaic agent and pass it through a filter, and these two individuals figured that out, if you put it in broth, it wouldn't grow. You had to put it on tobacco leaves. And for the foot and mouth agent, you had to put the filtrate onto animals. So that is a key concept, that not only these are small, but they need to reproduce in the host, which, as you see, is the definition of virus. They do not reproduce in broths. And the filters, the porcelain filters, had about a 0.2 micron pore size. But still, they were thought to be liquids, right? They thought, okay, these are different, but they're still liquids. They were actually called filterable viruses because we thought all viruses were infectious agents or bacteria, and then there were liquids that went through a filter. So we called them filterable viruses. And so if you look in the literature for many years, you'll find this term, filterable viruses, right? And here we have the first human virus, 1901, yellow fever, rabies in 1903, variola, it's smallpox virus, 1906, chicken leukemia, the first cancer virus, and polio, 1908, Rouse sarcoma, a cancer virus of chickens, bacteriophages in 1915, and in 1933, influenza viruses, but only after the 1918 pandemic. We didn't have virus isolated until many years later. Still calling them filterable viruses. The pace of discovery becomes more rapid. As you can see here, we're looking at years, and the cumulative number of discoveries for filterable viruses, protozoa, bacteria, fungi. So Koch pioneered being able to grow bacteria. And after that, the number of bacteria discovered exploded, right? Here we have the discovery of tobacco mosaic virus, and the number of viruses start to increase as methods for their study are detailed. But it wasn't until 1939 that we realized that viruses are not liquid. And the first experiment is this one, the electron microscope of the first virus particles. Now, Helmut Ruska was a German who built the first electron microscope in 1933. It's a wonderful technical museum in Munich where you can see it. It's a great museum. It's got all kinds of cool stuff. Even has an iron lung. And in 1939 was used to make the first photograph of a virus. And lo and behold, there were particles. Here's a bacterium with viruses attached to it, bacteriophages. They're particles. They're not liquids. Oh my gosh. So we stopped calling them filterable viruses. We just called them viruses because they were clearly different from bacteria, right? So up until now, we think they're different because they have this requirement for a host, but now we see that they're particulate, so we just call them viruses. Okay, let's do another. Which button do I push? I forget. Here we go. Another quiz. I have to go turn it on. This will be our last question. Which is a key concept first discovered about viruses that distinguish them from other microorganisms? They were too large to pass through a 0.2 micron filter. They could replicate only in broth. They made tobacco plants sick. They were small enough to pass through a 0.2 micron filter, or none of the above. So what is a key concept that distinguished them from other microbes and made them, just viruses, not filterable viruses? And let's see some some comments here. Breakthrough moments are awesome. Yeah, the discovery of particulate nature of viruses. So it's been less than 100 years since we actually identified viruses. It's 1900, a little more, right? Tobacco mosaic end of the 1800s, but not much. Not much. It's incredible. I have trouble imagining they thought viruses were liquid. Yeah, it's went through the filter. They knew bacteria were particulate, right? Because they could see them with Leeuwenhoek's microscope. And they figured something that went through was either small or was a liquid, yeah. All right, what do we have? We have 212 people who've logged into Socrative, so we're only at 98. I'm going to wait a little bit more. I'm going to look at some more questions here. Someone wants to work with me. I don't have people working with me anymore. I'm sorry. Okay, here's a good one. If some viruses can be seen in a light microscope, how come they were thought to be liquids? Well, you remember, the ones we see now, we didn't know back then. And first of all, the light microscopes weren't very good. And we only discovered recently the big viruses that we can see in a light microscope. So the ones that were worked on years ago, tobacco mosaic, the foot and mouth, etc., they were too small to see in a light microscope. We didn't have it. Are viruses solid? Well, they are, it's a good question. They're not completely full. So you have a shell, which can be empty if there's no nucleic acid in it, right? And then you put nucleic acid in, there's still spaces. So there is room for other, I don't know what molecules would fill the other space, but I wouldn't consider it solid like a chunk of glass is solid, no. And now I will remember to remove the comment. Does bigger genome always mean physically bigger virus? So, no, you could look back at one of the slides I showed you with the giant viruses, and you can see some of them have bigger genomes, but the particles are not as big as some viruses with smaller genomes, yeah. All right, let's have a look now at the quiz. For that, we go to the other view. Got 117 people. What did we get? So 84% of you. I have to say, having so many people answer is cool because in my class, I always have very few people, and you never know the trends, what they mean. Here, 84% of you said they were small enough to pass through a 0.2 micron filter. Now, here a lot of people picked other things. So why is this a bit confusing? So some of you said they were too large to pass through. The bacteria are too large, of course. The viruses pass through. That was the key. So that's not right. They could replicate only in broth. No, absolutely not. Viruses do not replicate in broth. They need a cell to get into, right? So that's not correct. 5% of you said they made tobacco plants sick. Now, that was clearly important for the first virus discovery, but it wasn't a key concept that they made tobacco plants sick. And given D, which is really a key concept, that takes precedent. And none of the above, a lot of you picked that, but it is D, right? That's the one that's important here. All righty, let's go back to our slides, and I'll get rid of me for this round. Some of you, oops, sorry about that. Some of you like to see me, some of you don't. We'll see how that works out over the next sessions. So aside from the electron micrograph, which showed that viruses were particulate, then we had the growth curve, which proved that viruses were not simply small bacteria. So two things in 1939, looking at them in an EM and this experiment, where again, if you put bacteria in a broth, they will start to divide by binary fission. If you put virus in a broth, they will not divide. You have to infect cells. And if you infect the cell, you have this substantial eclipse period over time during which you do not see the production of new virus particles. You simply do not see that with bacteria. Sometimes there's a bit of a lag in bacteria, but that's simply until the bacteria begin to make components so that they can split in two. The viruses have a substantial eclipse period during which you cannot find any new viruses made because the parts are being made. This was fundamentally different from bacteria. So the picture, of course, was very different. They looked very different from bacteria. But this mechanistically really nailed it, that viruses were different. They're not small bacteria. So now, post-1939, we call them viruses. And here's some electron micrographs of viruses just to show you how beautiful they are. We have in A bacteriophage with the lovely head where the DNA is. There's a tail and tail fibers. Then we have tobacco mosaic virus. This is the first discovered. It's a rod-like virus. We'll visit it many times. Rabies virus, bullet-shaped virus particles, and then a picornavirus, a round virus. Today we know many details about viruses. We know their structures at different resolutions, very high and intermediate resolutions. This is poliovirus at 2 different resolutions. You can see in the X-ray crystallography, which we will talk about later. You can see very high resolution, very less so with cryo-EM. We know the chemical formula for viruses as a consequence. And so as we have learned more and more, we have wanted to classify viruses, to study them. Humans like to put things in categories, even though no such categories exist in nature. And for many years, we used a variety of properties of viruses to classify them. You know, the kind of nucleic acid. Before sequencing, before genome sequencing, we would say, okay, is it RNA or DNA? What does the protein shell look like? Is there a lipid? What's the size? And then we would say, what kind of disease does it cause? What animals does it infect? We would use all of those things to classify. Nowadays, we mostly just look at the sequence. We sequence a new virus, and we immediately place it into the classification category. We don't really consider anything else than the sequence. And so the way we classify viruses is codified. You can find details at this website, which the ICTV is the International Committee for the Taxonomy of Viruses. It's an international committee that is charged with making the official names for viruses. And we use a hierarchical system that's used for other species, and we really go from order on down. We don't talk about kingdoms and phylums and classes. We have orders, which are collections of families, and then below the family is a genus, and then species. A species doesn't really exist as it does for, say, the mouse, Mus musculus. We know there's a species of mouse out there running around, but for viruses, because every isolate from everyone is slightly different, it's hard to really make a species, so we just use it as, in a classification sense. So orders end with virales, families with viridae. So Filoviridae would be the filovirus family. The genus, in this case, just has virus after it, and it would be the Ebola virus genus. And in this case, there are multiple species, and one of them is Zaire Ebola virus. There's Sudan Ebola virus, Reston Ebola virus. Okay, and it's one word for the virus names here in this case. And that's, of course, a filovirus, which we will talk about later on. Virus discovery now is not only driven by disease. As you can see, the very first viruses were discovered because they caused diseases in plants or animals or humans. Now we want to know what's out there, and this is the way it should be. We can easily sample many different sources, as we talked about earlier with the drones. We can get life forms, we can sample soil and water, sewage, and see what's out there. And here's an example from a study published in Nature a few years ago, where they collected 220 invertebrate species. Invertebrates, right? No backbones. Insects, crustaceans, nematodes, mollusks, cnidarias, all without backbones. And they ground them up and sequenced, and they found 1,445 new viruses. Never been described before. And the orange are RNA viruses, and these are all RNA viruses, sorry. And you can see they vary according to different species here, and some of them are high frequency and some of them are low frequency, but basically 1,445. The reason I show you this is that you could do this with almost anything and find new viruses. And you can, obviously, we do it with bats and we do it with rodents, but we don't do it as much as we should. And we actually don't know most of what is out there. We'll be talking more about virus discovery as we move on, for sure. Now, you may say, why do we care about any of this except what makes us sick? Well, there are a number of reasons. First of all, I think the biggest one is viruses outnumber cellular life by at least 10 to 1. They represent the greatest biodiversity on the planet. All right? More than anything else. There's more viruses than anything else that have genetic information. There's more diversity there. As I mentioned very briefly, they drive global biogeochemical cycles. Incredibly important for cycling of carbon and sulfur and phosphorus and many other elements. They shape host populations. I'm sure that hosts have been extincted by viruses in the past, and in turn the hosts shape virus populations. Many of them are likely to be beneficial. I think we need to find out why, because if they're beneficial, what are they doing, and can we substitute that? And finally, they're sources of new pathogens, right? Even though they may not currently be causing a disease, they could at some point in the future. So we should know what's out there. And we barely have any knowledge of what's out there. It's really sad. Now, you may be overwhelmed at the number of viruses, but we're going to put this in some kind of order. We're going to make an underlying simplistic view. And that's because of two things that we can use. First, they're all obligate molecular parasites that only function after they get into a cell. We could use that to unify all the viruses. And finally, they must make mRNA that can be translated by the host ribosome. This is a host ribosome. It looks a bit like a turkey, which was pointed out many years ago. This is a picture from the textbook, Principles of Virology, but it's a ribosome. All viral genomes have to make mRNA that can be translated by the ribosomes in the cell. Some viruses make parts of the translation apparatus, but none of them make the whole ribosome. And so they're parasites of the host protein synthesis machinery. We can actually use this to order all the viruses that are out there, and we're going to be doing that over the next 2 sessions. So next time we are going to talk about the infectious cycle. That'll be Monday. So come back. But meanwhile, we have some time to take questions. So let's do that. Let me look over here at the chat. Any known examples of beneficial viruses? Now, I assume you mean in people. No, I don't know of any. However, let's make this a little bigger. In mice, I think that norovirus is beneficial. It helps develop the gut. We talked about that at the beginning. It's very hard to do experiments to show beneficial viruses in humans, right? But maybe we'll get it someday. But no, not at the current time. Are viruses the only obligate intracellular parasites? They're small bacteria that require being inside of another bacterium in order to multiply. Those are the only ones I can think of. But they are different because they have ribosomes. They can do a lot of things on their own just given the right nutrients. And I think if you found the right conditions, you could probably get them to multiply outside of the cell. But you can't do that for viruses as far as we know. I'm taking away from this lecture that there are a lot of viruses everywhere. They're both good and bad. We just figured out they exist in the last 100 years. They're amazing and exciting. Yes, that's good. That's a good take home. And because of that, you should stick around for the rest of the course. And if you can't come live, I understand. Look, we had 417 people. If you can't come live, all the lectures will be saved on YouTube with the chat so you could go through it. Yes, bacteriophage are beneficial probably in us because they modulate our microbiome. I'd like to see more about the specific ways that they do that, but yes, that's a good point. Thank you. You're welcome. Doesn't virus replication in a host cell stop until the cell is dead? It stops when the cell dies. That's right. But sometimes it can go on for long periods of time, for sure. Oh, here's a good point. But phages will be beneficial for humans when they replace or complement antibiotics in our bacteria therapy arsenal. So we can use them. We can certainly harness viruses to be beneficial. And in fact, the last session of this course is called Beneficial Therapeutic Viruses. We're going to talk about the ways we can harvest viruses to do the good things for us like that. Yes, absolutely. Wouldn't viruses used to create and deliver vaccines be considered beneficial? Yeah, that's because we've modified them, right? So I'm thinking about naturally occurring beneficial viruses and then the ones we modify that are beneficial. Absolutely. I want to know if any of the viruses that just infect us without any modification, all the herpes viruses, the polyomas, what do they give us? That we don't know. But absolutely, all the viral vectors that we use for gene therapy, for vaccination, totally agree. They're beneficial. Yeah. Why are there no negative sense single-stranded DNA virus? So most of these single-stranded DNA viruses package either strand. It doesn't matter, right? Because if you have one strand, you can make the other. So they exist, but they're always a mixture. Have warm, fuzzy learning stuff feelings. That's great. I'm glad. So was this a successful experiment? I hope so. I think there's some things that could be done better. I don't. The monitor that I'm looking at for the chat questions is too far away. I can't see it. So I have to move it closer or get a bigger monitor. This is a pretty small monitor. It's 15 inches. So I got to fix that so I can see the chat questions better. And if you have other issues, let me know because I want to fix it. This is an experiment, right? Could you remember at the top of your head if you and Nels discussed papers on infections and their implication for human evolution? Yeah, I don't remember the episode, though. But there aren't that many. You could easily scan through and find out which ones. Are viruses found in placentas? Well, viruses can certainly cross the placenta, sure. That's the ones that cause congenital birth defects. So, yes, not every one. The placenta is a good barrier that keeps out most viruses, but a handful of them can get in, including Zika virus and cytomegalovirus and others. Can we synthesize a virus? What we can do is synthesize the genome of a known virus and put the genome into a cell, and out will come virus. We can't make a brand new one. Humans aren't good enough to do that. Only nature is good enough. The Russians have used phages for years. Yes. In fact, shortly after phages were discovered, remember 1915 or so on my slide? Shortly after they were discovered, the Eliava Institute was founded in Georgia, and that exists to this day to use phages for bacterial therapy. And there are some very interesting advances in that, which we've talked about on TWIM and TWIV. Yep. All right, so here's an interesting one. Are you calling an unlive virus an organism because it has organization? I thought organisms were alive. So an organism with two phases, right? Because there's the infected cell, which is clearly alive. The particle is not an organism technically, but the it is an organism with two phases. That's what I mean. I hope that's clear. If a virus has a fat layer of protection, it is disarmed with chemical interaction. I was curious of natural processes of a bay leaf used in food. So you can remove the infectivity, destroy infectivity of viruses by agents that destroy the lipid bilayer, the membrane, like ether and other things that are detergents. Soap will trash viral membranes. And that's once the membrane's gone, then it loses infectivity. TWIV followers since the beginning, a couple of aborted attempts to complete your course. Now's the time. Oh, here you go. You know, this gives you some motivation because there's a chat. There are a lot of like-minded people here that you can chat with, so maybe that helps. Let's see. Someone is asking about the quizzes. So I don't get, I get, you know, how everyone does, but there are no names associated with it. That is a preference that I can set in the beginning, and I don't need to know your names. In fact, there's no way I have. Yeah, they're all anonymous. I have to explore whether there's a way to get your own. I don't think there is, though. But we're going to have other quizzes where you can evaluate your own process. Okay, so that'll probably, David, serve your purpose there. Can viruses hibernate? Well, I don't know if hibernation is the right word, right? Hibernate is, you know, we usually reserve that for animals that, you know, are still metabolizing. So what viruses can do is remain infectious but not infecting a cell, as long as if they're frozen, for example. So not we, but virologists recovered viruses from Siberian permafrost by simply getting the ice and thawing it. So it lasted for 30,000 years. So it can be done, and as long as they're frozen under the right conditions, they will retain infectivity. Yeah. How does infecting RNA produce mRNA? Well, you're going to learn about that in a couple of sessions. We have a whole lecture on that. Stay tuned. Why would viruses have a protein but not lipid? Because in many cases, a protein coat is sufficient to protect the genome, and then the protein coat can be recognized by cell receptors. It works to protect the genome, which is a big function, and to get the virus into a new cell. Can viruses use the components of newly dead cells to replicate? Well, if those components could get into the virus-infected cells, then yes. And that's how we think autophagy works, right? If a cell is dying, vesicles form in the cytoplasm that grab components, so they leave the cell and they bring it into other cells. So yes, they could, so as long as they got into another cell, right? That would be the key. How are virions assembled in cells? Random collision. So we're going to have a whole session on assembly, right? But in the protein sequence, we have the information for the protein to fold and interact with each other. So it's basically random, yes. So someone says we need to up the volume. Well, I can do that. But I think some of you already have reasonable volume, right? You've already said it's good. So I'm going to turn it up here a few notches. So I just cranked it up a bit. Let me know if that's good or if it's too much. I will go on to the live chat and see. All right, so then this is a white background, right? I'm going to put something here. I commissioned actually a nice virus print. I'll probably move away from the wall so you can see more of it. I kind of miss my office with all the virus stuff, but you think it's good to have it clean, you know? Well, maybe. See, look at this. So someone likes it and someone else doesn't like the white background. So you you can't please everyone. That's fine. My problem is I try and please a lot of people. Andrew Kiora. Yes, I think you're going to be involved because of the chat, right? So you got a lot of like-minded people, as I said. Some words need to be clarified. Yeah, I know this is a problem that I can't always look at the chat, right? Maybe I could figure out a way to do that. But I will get distracted because a few times you may have noticed I've paused, and that's because I saw a question in the chat like your camera should be moved up, right? And that totally distracts me. I'll figure something out. Enteric having to do with the gut, of course. Yeah. Video quality isn't as good. Well, it looks really good here. You want more vibrant colors? Yeah, I could fix that. But the content is what's key here, right? And some people want a green screen. Okay, all the classes are 11 a.m. Monday and Wednesdays, okay? See you next time. I'm not leaving yet, but I'm just going through some of the questions. Yeah, I am just this is like the first week at the incubator, right? And it's not honed yet. It will be fixed. Everything will be fixed. Like, it is too bright. I totally agree, but I will get it fixed. Don't worry about it. Are some people sensitive to the pepper virus? I don't think it's the virus. It's probably the capsaicins in the pepper that are the spicy components. The virus is not reproducing in you. The only possibility is that it might induce some reaction, but I really think the peppers, the spice themselves, the capsaicins are doing that. Interesting idea, though. There's a lot of them. Thank you, Mr. OzzyCam, for your support. You can always support us. Maybe you feel it's weird to support a lecture. Well, come to the live stream tonight at 8 p.m. We do this at 8, which is a totally different thing. Me and Amy Rosenfeld answer COVID questions, 8 p.m. And if you have COVID questions, bring them. Same place. And I do think having it live is cool. And I can hopefully do better at answering your questions, right? Oh, Discord channel. I have set up one. I haven't had a chance to sort it out. Someone actually volunteered to help me sort it out, and I have to get back to them. So, yeah, we're going to have a Discord where 24/7 you could go and find people, like-minded people, moderated. Yeah, I want to do that. I think that would be cool. Yes, Georgia, the country in the former Soviet Union, now the independent country, Georgia. That's where the phage therapy work is being done. Are there any resources for learning about the ancient history and references to disease that you would recommend? So there's a book by Michael Oldstone, Viruses, Plagues, and History, which gives a good overview of the role of viruses in history. Viruses, Plagues, and History by Michael Oldstone, right? We could actually find this. Let's get it up. Viruses, Plagues, and History. Viruses, Plagues, History. Hey, there it is. Let's see. What number is that? Gosh, I just forget. I'll take off the question. There it is. Viruses, Plagues, and History, Past, Present, and Future. That's a good intro to what you're asking about. Oh, we have someone from Malaysia, 1 a.m. Yeah, it was hard for me to get a time that would work for global. So Asia is going to be the one that's going to have a problem. Do viruses have a hard or soft limit to their evolution? You mean how much they can change? We're going to actually talk about that. There are a lot of different potential combinations of new genomes, but we rarely see them. So there are things that can't change. Nucleic acid doesn't change from DNA to RNA or vice versa. The capsid doesn't change. So there are some hard limits. But within the boundaries, you can do a lot for sure. Can the eclipse period be considered the same as the pre-symptomatic phase? It's totally different, right? Because in the pre-symptomatic phase, virus particles are being made. Whereas in the eclipse, it's in a cell before virus particles are being made, okay? Yeah, Arrowsmith, one of my favorite books, phage therapy, absolutely. For sure, phage therapy was featured in that book, yeah. Do you believe viruses follow bacteria and archaea on the evolutionary scale? No, I think, as I said, I think nucleic acids were their first self-replicating nucleic acid. They entered cells. But we're going to talk about that in the evolution lecture. So stay tuned for sure. Did the mic gain help? Let's see if some people answered me. Who knows? I will have to go back and look. Video is better in my home. Well, it's different, right? We'll fix everything. Looks like the audio is better. Thank you for your contribution. I really appreciate your contribution. Oh, is there an RNA repair mechanism? Absolutely. The coronaviruses have it. Actually, they're the only family that have it, and we're going to talk about that. And that's why their genomes probably get too big. Absolutely. Shout out for COVID-19 Q&A with A and V. That's tonight, 8 p.m. YouTube. If you can handle 4 hours of virology in one day, right? Well, Bunny, thank you for your super sticker. I really appreciate it. I want to thank all the moderators too before I leave. Mr. Ozzycam, Vanity Nutrition, Les Fabi, Steph, and one other person whose name is evading me. How much virus research has been done in household pets? Do you mean in people's pets, understanding what they have, or using them as laboratory animals? So you don't do experiments in people's pets. You can diagnose what infections they have and treat them. But there are places that do research in dogs and cats. For example, up at Cornell, there's a really well-known canine virus research institute where canine parvovirus vaccines were developed and so forth. So quite a bit is done. Can a bacteriophage be modified to infect human cells? What a good question. I think you could modify it so that it would attach and get in, but I'm not sure it would work because all the signals, all the regulatory signals are different. So it would have a hard time reproducing. Thank you for your contribution. If you get the flu shot, does the enhanced immune reaction help prevent viruses? So you snuck in a question with your contribution. Some people think it does, and other virus vaccines as well. Super chat. Thank you, BJR2, for your contribution. Thank you, Amos. Really, really appreciate it. Nathan, thank you for your contribution. So I'm moving into a phase where I'm going to be here at the incubator most of the time doing podcasts and teaching. It's what I like to do for the rest of my life, actually. My career after Columbia. And there'll be a time when I don't teach my Columbia course anymore, but I'll continue to teach this. And I have plans for other courses, folks. If this works out, meaning people like it and they come, I'll do other courses like specific viruses, like emerging infections, like a course on pandemics. Can do all kinds of stuff. Really appreciate it. And I love that people are all over the world. Is this every Monday and Wednesday at 11 a.m. Eastern Time? That's right. All right, should wrap this up. I promised you 2 hours. So let's just thank people. Thank you, Hotel Papa 100. Really appreciate your support. Oh, I like Adam. You look great, younger even. That's the reason I bring this up. Thank you for saying I look younger. The content always suppresses. Yeah, but I know that you need to have a good image. Really, really know that that's important. Thank you, Marty, for your contribution. I recognize you from the live stream. Thank you for coming and for your support. Thank you, Riri Med from Baltimore. And I think some more super chats coming in. Thank you so much, Susan. Your support is appreciated. Of course, as I said, this whole incubator, and at some point when it's set up, I'll have another camera so you can see my environment. It's all paid for by your contributions. And, I did a lot of podding before contributions, but kind of in another level now. I will still always do it, but your contributions really help. So the Q&A with A and V will not be from here because it's 8 p.m. and I don't want to be here in New York at 10. The trains are like an hour apart, and if I miss the 10:12, then I have to wait till 11:12. So that'll be from home tonight. Yeah, for sure. Thank you, Ralph, for your contribution, and Kate, thank you for your contribution. All right, that'll do it for first lecture. I think it's a success. Come back on Monday, 11 a.m. Eastern. And remember, there's going to be a quiz posted tomorrow. You can find it at the website. Thanks for coming, everybody. See you next time.